imatinib plus savolitinib for patients with EGFR mutant MET amplified and non-small cell lung cancer after progression on prior first and second generation EGFR inhibitors and also third generation EGFR inhibitors. Dr. Sequist.
Thank you, Dr. Yap. Thank you to the organizers for allowing me to give this presentation. On behalf of the TATN investigators, I'm pleased to be here. Thank you. So as mentioned by Dr. Yap, if you look in your program, there are two sequential abstracts, and because they cover two arms of the same study, we're going to be combining them into one presentation with the permission of AACR. My Twitter handle is on the bottom right-hand corner of this slide, and you'll see it again on the conclusion slide at the end. If you don't have a chance to ask your question at the microphone today, I'd be pleased to hear from you in the coming days on Twitter. Here are the disclosures for myself and my co-investigators. So over the last 15 years, through various types of research, both clinical and preclinical, we've gotten a good picture of what causes resistance to EGFR TKIs among EGFR mutant cancers. We have seen from clinical repeat biopsy studies that up to 10% of patients who are progressing on first or second generation EGFR TKIs and up to 25% who are progressing on third generation TKIs will have MET amplification driving their resistance. Preliminary, more recent studies looking at ctDNA in the resistant population from large randomized trials, including FLAURA and AURA3, have shown us that MET-driven resistance is a big player for osimertinib resistance as well. Keep in mind that these ctDNA-based studies may have an underestimate of the percentage of MET that is driving resistance because not all of the tumors are shedding ctDNA. So these clinical studies really bolster our preclinical understanding of MET amplification in these cancers, which goes back to 2007 when Jeff Engelman published a seminal paper in Science first describing that MET amplification could be a resistance mechanism. And importantly, and relevant to this presentation, in that paper, he showed that in the preclinical models, when you had an EGFR mutant cancer that was resistant because of MET amplification, neither a MET drug alone nor an EGFR drug alone was sufficient to cause cancer regression. You really needed the combination in order to be active. We have seen since then a handful of case reports, single patient case reports, looking at that strategy of combining a MET inhibitor and an EGFR inhibitor. So this study will focus on osimertinib plus savolitinib. You've already heard in this session a bit about savolitinib. It's an oral potent, highly selective MET TKI, which has preliminary anti-tumor activity in combination with EGFR TKIs in such patients as I described, EGFR mutation positive with MET-driven resistance. Today I will show you updated interim data from two cohorts of the TATN phase 1b trial, which is osimertinib plus savolitinib in EGFR mutant patients with known MET-driven resistance. We'll first look at a cohort of such patients that developed their MET-driven resistance on first or second generation drugs, and then we will look at a cohort who developed MET resistance on third generation drugs, including osimertinib. Critical is that MET-driven resistance is not necessarily easy to detect or standardized at the current time. MET pathway activation can occur through a diverse array of mechanisms, and like any acquired resistance mechanism, there can be heterogeneity between tumor sites, even within a single tumor, and there is essentially no standard test. Fluorescence in situ hybridization, immunohistochemistries, next-generation sequencing, and real-time PCR have all been used to assess MET pathway activation, but at this time there's a lack of consensus about which test is the best and what is the optimal cutoff value. Comparative analyses were done on the 250-some patients that screened for TATN, and if you're interested in looking at the comparisons between the different MET detection methods, I urge you to go to poster board 20, abstract number 4897, which is going to be on Wednesday morning, presented by Dr. Hartmeyer. One of the takeaway, or a couple of the takeaway points from his poster are that MET polysomy was largely missed by NGS testing, and while MET IHC 3+ staining overlapped extensively with MET FISH, there was also an additional population picked up by the IHC. For TATN, enrollment was allowed and treatment was initiated for many patients based on local testing. If local testing wasn't available, they could utilize central testing, but local testing was allowed in this study by either FISH, NGS, or IHC. All of the patients had to have a tissue biopsy done after progression on the most recent prior regimen. So these tissue biopsies are done at the time of study enrollment. Cytology was not allowed, ctDNA was not allowed, and for the patients to be labeled as MET positive, they could either have by FISH, MET gene copy number greater than or equal to 5, or MET to CEP7 ratio greater than 2. They were going to use a local NGS panel. It should include a minimum of 20% tumor cells and 200-fold depth, and the MET signal should be at least 5 copy numbers over the tumor ploidy. And for immunohistochemistry, the staining had to be 3+ or greater, 3+ in 50% or greater of the tumor cells. An overview of the TATN study is provided here. So all of the patients on TATN were EGFR mutation positive who had progressed on at least one prior TKI. The study started off looking at dose escalation and dose finding of 3 different combination regimens, all based on osimertinib. There was osimertinib and savolitinib, osimertinib and selumetinib, and osimertinib and durvalumab. The durvalumab arm, as you may recall, was halted during dose escalation due to unexpected excess toxicity. This was reported in 2016. The other 2 drug combinations went on to Part B, which were dose expansion cohorts. Today, I will be focusing on 2 of the dose expansion cohorts within the osimertinib savolitinib study. Patients on both of these arms were MET positive, as mentioned. After my talk, you will hear from Dr. Ramalingan, who will be talking about the second row, osimertinib and selumetinib. The TATN overall study also includes a Part C, which is a dose-finding study in Japan, as well as a Part D, which is looking at alternative doses of savolitinib, but results from Part C and Part D will not be presented today. Okay, so turning to the savolitinib arms. These 2 expansion cohorts included adult EGFR mutation positive patients who had progressed on at least one prior TKI with a good performance status, and as mentioned, they were all biopsy proven to show MET as a driver of their resistance at the time of going on study. One cohort that we'll be talking about received or developed their MET-driven resistance while on a 1st or 2nd generation TKI. They were known to be T790M negative as well. And this cohort and the results from this cohort will be color-coded in green for the rest of the presentation. Another cohort received prior treatment with a 3rd generation TKI when they developed their MET-driven resistance. This cohort will be coded in blue. Both cohorts received the same dosing, the same treatment regimen, osimertinib 80 milligrams a day, savolitinib 600 milligrams a day. This was the dose that was determined from the dose finding Part A of the study. Both of these cohorts had a primary endpoint of safety and tolerability, and efficacy endpoints were secondary outcomes. So the green-colored cohort first, which is the patients who developed MET-driven resistance on a 1st or 2nd generation drug and were T790M negative. The demographics of this patient population are shown here. The cohort was 46 patients in size. You can see that 80% of the patients were Asian, and the majority, 67%, had had only one prior treatment. So this is essentially second line. The patients had received the one prior treatment was their 1st or 2nd generation EGFR TKI. And rather than developing T790, they had developed MET amplification. Following the patient disposition, 46 patients were assigned and received treatment. 25 patients had discontinued both osimertinib and savolitinib, and 6 had discontinued savolitinib only. The most common reason for discontinuing osimertinib was progressive disease. The most common reason for discontinuing savolitinib was adverse events, and 15 patients continue on treatment still. Looking at the adverse events, we can see overall that 98% of patients had any adverse event, 54% of patients had a grade 3 adverse event. And looking at the adverse events that led to dose reduction or discontinuation of drug, you can see that that was more common for savolitinib compared to osimertinib, but overall was a minority of patients. What types of adverse events were most commonly seen? Well, the most common were grade 1 or 2 nausea, diarrhea, fatigue, decreased appetite, and pyrexia. Seeing grade 3 or higher events of this nature was uncommon. Looking at the serious adverse events that were causally related to treatment, overall, these were low. There were a handful of patients who had a hypersensitivity type reaction to the combination, and this was usually preceded by pyrexia or fever. Looking at the preliminary anti-tumor activity for this cohort, we can see that among the patients who developed MET resistance after first or second generation TKI, the objective confirmed response rate was 52%. There are 16 patients currently categorized as stable disease, and this includes 4 with unconfirmed partial responses. When looking at the waterfall plot, recall that all patients had to be MET positive on some test, either local or central, in order to start treatment. The plus signs below the bars denote the patients who had central confirmation by FISH test positive for MET. A common reason not to have central confirmation FISH test positivity was inadequate tissue to undergo the central confirmation. The median duration of response among this cohort was 7.1 months. So now we'll turn our attention to the other cohort of patients, those EGFR patients who developed MET-driven resistance while on a third-generation EGFR TKI. The patient demographics for this cohort of 48 patients are listed here. Again, the majority were Asian, and I draw your attention to the number of prior therapies as it differs from the other cohort. In fact, only 1 patient here had received a third-generation EGFR TKI in the frontline setting and then went on to this trial in the second-line setting. 27% of patients had had 3 prior lines of therapy, and another 27% of patients had had more than 3 lines of therapy. So a common clinical scenario for patients entering this arm of the study would be that the EGFR patient would receive a first or second generation TKI in the front line, go on to develop T790M, then receive a third generation TKI, and then the resistance to that drug would be driven by MET. You can see all patients had received a prior third-gen drug, and over 75% of them had received osimertinib, either alone or sequentially with an experimental third-gen TKI. 48 patients were in this cohort and received treatment. 31 patients have discontinued both drugs. There were no patients who only discontinued one or the other in this cohort. The most common reason for discontinuing drugs was progressive disease, and 17 patients remain on therapy. Looking at the overall adverse events, 98% of patients had experienced an AE and 38% a grade 3 or greater AE. You can see that the adverse events leading to dose reduction or drug discontinuation were generally lower in this cohort compared to the first cohort that we examined. The types of adverse events that were seen were very similar to the other arm. It's primarily grade 1 and 2 GI type of side effects like nausea, vomiting, diarrhea, as well as fatigue. Grade 3 events of this type were uncommon, but serious adverse events possibly or causally related to treatment, again, were uncommon, but did include some rare cases of hypersensitivity type reaction. The anti-tumor activity of this cohort is depicted here. The objective confirmed response rate is 25% at the current time. 21 patients are designated as stable disease, including 5 patients with unconfirmed partial responses. And again, with the waterfall plot, recall that all patients were MET positive, and the plus sign denotes those that had central confirmation of MET FISH positivity. The duration of response on this cohort was 9.7 months. So in conclusion, these 2 dose expansion arms of the TATTON study show that osimertinib plus savolitinib has an acceptable side effect profile, and the combination shows encouraging anti-tumor activity in EGFR mutation-positive patients with MET-driven resistance who are progressing on first, second, or third generation EGFR TKIs. Our data suggests that the combination of osimertinib and savolitinib could overcome MET-driven resistance mechanisms, but further research is needed to determine the final effectiveness of this therapy. And to that end, there are 2 studies which are either starting or ongoing. The SAVANNAH study has just begun. This is a single-arm phase 2 study looking at osimertinib and savolitinib in patients who have previously received osimertinib in the first, second, or third line setting. Importantly, this study will help us look more specifically at the different ways in which one can detect MET-driven resistance and see what the differences are and the similarities between these methodologies so that the optimal biomarker testing methodology can be determined. The primary objective of the SAVANNAH study is to look at response rate. In addition, the ORCHARD study will be opening sometime in 2019. This is a multi-arm study looking at acquired resistance to frontline osimertinib, specifically frontline. And there's many arms that will be part of this study, some of them biomarker matched, some of them non-targeted or non-biomarker matched. But for patients who are found to have MET amplification after first-line OC, they will be shuttled toward the osimertinib savolitinib arm of ORCHARD. With that, I would like to thank all the patients and their families that have participated in TATTON, as well as the staff and investigators at all the study sites, the team at IMEDCOMS for their help, and this study was sponsored by AstraZeneca. Thank you.
Thank you very much. Any questions?
A question from the discussant.
Yeah.
Roy Herbst from Yale. I'll discuss later. I'm just wondering, some of your deepest responses in one of your waterfalls were patients that were not confirmed by FISH. So I was wondering, is it just that there wasn't sample for that, or did it not meet the criteria, and how do you explain that going forward?
So it's a mixture, and I don't have the data broken down for each specific line on the waterfall plot, but I can tell you from the patients that we enrolled at Mass General, many of them did not have sufficient tissue to go through the testing centrally at the sponsor, but were highly MET amplified, too numerous to count on primary testing done at home.
Thank you.
Yeah.
Any other questions? Can you tell us a bit more about the anaphylactic shock?
Yeah, so this is something that we're still learning about. It happens rarely, but it does seem to happen in both arms of the study. It may be more common in Asia, and as you recall, a large portion of the studies of the participants here were Asian, but there seems to be some sort of hypersensitivity syndrome that's often has a prodrome of fever or pyrexia, and then patients may have more severe reactions, especially if they stop the drugs and are re-challenged. And so there's been a lot of adjustments made to the study and the protocols on how to best handle these toxicities, including trying not to stop drug if you intend to keep going, and prophylaxing with steroids or antihistamines.
Great. Okay, in the interest of time, we'll move on. Thank you very much.
Thank you.
So I'd like to invite Dr. Suresh Ramalingam from Emory who will discuss more results from the phase 1b TATAN study. This time round, osimertinib plus selumetinib for patients with EGFR mutant non-small cell lung cancer.
Good afternoon. Dear colleagues, on behalf of my co-investigators, it's my privilege and honor to present the results with the combination of osimertinib and selumetinib in patients with EGFR mutated non-small cell lung cancer. This cohort was evaluated as part of the TATAN study, which you have just listened to the presentation of Dr. Sequist about the MET cohort specifically. These are our disclosures. It'll take the next 20 minutes to read it, so I will move on. Targeting EGFR tyrosine kinase resistance mechanisms is of key interest as the newer generation of EGFR inhibitor osimertinib has moved to the frontline therapy for patients with metastatic non-small cell lung cancer. For patients with frontline first or second generation EGFR TKI, T790 is the most common resistance mechanism. With the movement of osimertinib to the frontline setting, we're learning about newer mechanisms of resistance that are specific to that setting. In all, upregulation of the RAS-RAF MAP kinase pathway seems to be central to mechanisms of acquired resistance to EGFR inhibition. And combining targeted therapies, which could not only inhibit the EGFR tyrosine kinase, but also some of the resistance pathways, could be a way to either avoid or overcome acquired resistance. With these in mind, the TATAN study was specifically designed to look at specific combination approaches. In this situation, we will talk about the combination of MEK1/2 inhibitor selumetinib with osimertinib for patients who developed acquired resistance to prior EGFR tyrosine kinase inhibition. Now, selumetinib is an orally administered potent inhibitor of MEK1 and 2. It has a relatively short half-life, and that's why it's given more than once a day, often in studies. It has been studied in non-small cell lung cancer in different contexts. One was in patients with KRAS mutated non-small cell lung cancer, where selumetinib was given in combination with docetaxel. Now, despite a promising phase 2 study, the confirmatory phase 3 study with this combination of docetaxel and selumetinib failed to demonstrate efficacy. Selumetinib is now under investigation in pediatric tumors, specifically with neurofibromatosis in patients with a specific form called plexiform neurofibromas, where it has shown very promising anti-cancer activity with 72% response rate. So this compound is active and is of considerable interest in studying patients, in the setting of acquired resistance. So the design of the TATAN study with selumetinib is noted here. We have 3 specific cohorts here. Cohort 1 studied selumetinib in combination with osimertinib, and the dose of selumetinib for this cohort was 25 milligrams twice a day. This specifically was done in Asia because prior studies had shown that patients from Asia have higher plasma exposures to selumetinib, and therefore we started with a lower dose. For the patients from the rest of the world, we used selumetinib at 50 milligrams twice a day, with osimertinib at its approved dose of 80 milligrams a day. And the third cohort we investigated was an intermittent schedule of selumetinib at days 1 and 4 of every week of therapy at the dose of 75 milligrams twice a day. Now, each of these dose cohorts had dose escalation built in, except that for the intermittent cohort, we studied the same dose of 75 milligrams twice a day of selumetinib, but it was with 4 consecutive days on and 3 days off of therapy each week. Once we identified the optimal regimen for further development, our plan was to evaluate the safety and efficacy of this combination in an expanded cohort of patients, and I will show you the results from that as well. So the primary endpoint for this trial was to determine the dose-limiting toxicities and to define the recommended phase 2 dose for the combination of osimertinib and selumetinib. The secondary endpoints included determination of the objective response rate, duration of response, and drug-drug PK interactions. We included patients who were older than the age of 18 in the rest of the world and 20 or greater in Asia, in Japan specifically. patients with advanced stage non-small cell lung cancer bearing an EGFR sensitizing mutation and an acquired resistance mutation in the form of T790 were eligible. Patients were required to have only the native mutation. If they had T790, they were eligible to come in. They should have received prior therapy with an EGFR inhibitor, and the performance status was ECOG 0 or 1. We excluded patients that had received selumetinib or any other MEK, RAS, or RAF inhibitor. So for the dose escalation part, the patient demographic characteristics are shown here. There were a total of 36 patients. 53% were women. And specifically, I want to talk about the number of prior regimens in these patients. As you can see, the median was 3, and 6 patients had received prior T790M-directed therapy. When you look at the baseline EGFR mutation type, exon 19 and exon 21 were seen in 26 and 8 patients respectively. 28 patients had T790M mutation. Patient disposition is summarized in this slide for each group. 13 patients were treated in the selumetinib continuous Asia cohort, 12 patients in the continuous rest of the world cohort, and 11 patients in the intermittent cohort. Of these patients, 1 patient is still on therapy on the Asia cohort, 1 patient is ongoing on the rest of the world cohort, and there is 1 patient who is on treatment in the intermittent group. In every one of these groups, treatment progression or disease progression was the most common reason for discontinuation of study treatment. This slide summarizes the adverse events noted for patients in the dose escalation group. And as you can see, that when you look at the overall toxicities of these regimens, specifically focusing on grade 3 or 4, in the osimertinib Asia cohort, we saw a high incidence of grade 3 toxicities, particularly in the higher dose cohort. In the rest of the world group, once again, we saw a grade 3 or 4 group. The intermittent group appeared to have a lower incidence of grade 3 or 4 toxicities. When you look at toxicities that resulted in discontinuation or dose reduction, it was more commonly done for selumetinib than with osimertinib. And in the intermittent group, about 18 patients had dose reduction, 5 patients had dose reductions for osimertinib and 6 patients for selumetinib. Dose-limiting toxicity data are summarized here. In the intermittent group, there was no dose-limiting toxicity, and that was part of the reason why this regimen was chosen for further evaluation in Part B. In the rest of the world continuous group, we saw 4 dose-limiting toxicities: diarrhea and nausea, and pneumonitis in the higher dose level. In the 50 milligram BID, we did not see any DLTs. For patients in the Asia cohort for continuous administration, there was 1 dose-limiting toxicity at the 50 milligram dose, and that was in the form of diarrhea. For the 25 milligram dose, we had 1 DLT in the form of ALT and AST elevation. This slide shows the preliminary anti-tumor activity in the dose escalation cohort. As you see here, the overall response rate was 42%. For patients who had received a prior third-generation EGFR TKI, the response rate was 17%. Among patients who had not been treated with a prior third-generation drug, for T790M positive disease, the response rate was 57%. For T790M negative disease, the response rate was 14%. The swimmer plot here shows that the responses were durable. The median duration of response was 16.6 months. And for the responders, 77% of the patients remained progression-free at 12 months. Pharmacokinetic data, top line, are summarized in the slide here. You see the osimertinib PK characteristics and the selumetinib characteristics. And what you can conclude from this is, regardless of the dose of selumetinib that was administered with osimertinib, its PK characteristics are unchanged. With selumetinib, there is a dose proportional increase in exposure, and there was no suggestion that the PK characteristics of selumetinib were different when it's given in combination with osimertinib compared to historical controls with monotherapy. So there were no potential drug-drug interactions in this study. Now I will talk about the expansion cohort, where we chose the intermittent regimen for further evaluation. In this cohort, patients received osimertinib at the dose of 80 milligrams per day and intermittent selumetinib at 75 milligrams twice a day for 4 days on, 3 days off every week. 47 patients were included in this group. Patient demographics for these 47 patients are summarized in the slide. 62% were women. Number of prior therapies, about 17 patients, or 36%, had 3 or more prior regimens, and 17% had come into the study after just first-line therapy. EGFR mutation type is noted below. T790M mutation was seen in 57% of the patients, and exon 19 deletion was seen in 64% of the patients. Of the 49 patients, 12 had received prior first and second generation TKI, and 35 had received prior T790M directed third generation tyrosine kinase inhibition. In this group, 1 patient is still ongoing study treatment. In the prior first and second generation group, there are 4 patients who are receiving both drugs, and there are 2 patients who are receiving osimertinib alone. And once again, disease progression was the most common reason for treatment discontinuation. This slide summarizes the anti-cancer activity noted in the expansion cohort. The overall response rate for the entire group of 47 patients was 34%. For patients who had received a prior first and second generation drug, the response rate was 67%, or 8 out of 12. For patients who had already been exposed to a prior third generation EGFR inhibitor, the response rate was 23%. These responses were durable. The median duration of response was 9 months, and patients remaining at response at 12 months was 31%, and at 18 months, 25% of the patients with the response were still in a state of response. And you can see here that the responses were seen in both groups of patients, regardless of prior T790 inhibition. Summary of adverse events are noted here. These are all-cause adverse events. Grade 3/4 events were noted in about 40% of the patients, of which 21% were thought to be treatment-related. Toxicities leading to reduction in dose or interruption of the dose were seen. 23% for patients with osimertinib, 34% for patients with selumetinib, and these were lower for patients who had already been exposed to prior third-generation drug, which would be something that one would expect. These are the most common adverse events. Majority of them were grade 1 or 2 in severity. Grade 3 events were relatively low. Diarrhea was the most common grade 3 adverse event noted for this group of patients. The rest of the toxicities include diarrhea, decreased appetite, stomatitis, paronychia, and so forth. When we specifically look at treatment-related toxicity, once again, diarrhea is seen in patients, 6 patients or 13% for patients who were treated with the combination. The rest of the toxicities are primarily grade 1 or 2 in severity. So in conclusion, the schedule of osimertinib plus intermittent selumetinib was chosen as the regimen with the most favorable therapeutic index from this experience. We conclude that combining osimertinib with intermittent selumetinib is feasible with manageable toxicity and has already demonstrated promising preliminary anti-cancer activity. There were no drug-drug pharmacokinetic interactions. We are conducting analysis to explore the correlation between baseline tyrosine kinase resistance mechanisms and response to this combination approach. And finally, there is an ongoing study at Dana-Farber Cancer Institute that looks at this combination in newly diagnosed EGFR-mutated non-small cell lung cancer patients. We would like to thank the patients, their families, and all research staff and investigators that participated in this study. A special thank you to Dr. Karthik Vishwanathan for his help with pharmacokinetics. And I want to thank you for your kind attention.
Thank you very much. We're open to questions. Selumetinib has got a pretty short half-life. Do you think it can push the dose and schedule further beyond 4 and 3 off?
I think there is room to further refine the intermittent dose schedule. I would say that the continuous dosing, pushing the dose is probably not a good idea.
Agree. Okay, I think there's a question.
That was a great presentation, Suresh. I don't know if I missed or misread one of these data points. You mentioned that in one of your initial cohorts, there's a patient, the range of chemotherapy regimens was 1 to 13. Was there a patient who received 13 prior regimens before being treated with this?
Well, with EGFR mutated disease, as you know, the survival, thankfully for these patients, is now measured in years, not in months. So there is more and more patients that we're seeing that have received multiple prior regimens.
Okay, great. Thank you very much.
Thank you.
I'd now like to invite Dr. Roy Herbst from Yale to discuss the science behind the study.
Thank you very much, Dr. Yap. Thank you to all the presenters. I was given an interesting title, Can the Challenge of Non-Small Cell Lung Cancer Resistance Be Met, or Will We Not Mech It? That's what I was given. So if you stay till the end, and I hope you will, I will answer that question in the final slide. And here are my full disclosures. So what I'll do today are 4 things. Review the current standard of care for non-small cell lung cancer, mutated EGFR disease, analyze the efficacy and safety results, as we've just heard in the last 2 presentations representing 3 abstracts, have a few outstanding questions I'd like to bring up. How can we get as close to a cure as possible in EGFR mutated disease? And then what is the impact of all these data on clinical practice? What are the next steps? So what about EGFR mutated lung cancer? Well, we've come a long way. We've heard, this has been a very international presentation. In fact, most of the patients we heard about today were treated in Asia. So if you include the entire world, there are a lot of cases of lung cancer, over 2 million. You can see in the US, we have over 200,000. Most of it is non-small cell lung cancer. But if you look on the right, we now are in the era of targeted therapy with many potentially actionable mutations. And it's so nice to see that MET is now getting its due. The literature would tell you that it's 7%, but it's really much more. And now I think we have evidence from the data today, and more will be generated, that it could be as high as 15%, 20%, or even 30% in patients in this new era of resistance to first, second, and now third generation EGFR inhibitors. Now, there's a long history of EGFR. This is a timeline article written quite a while ago, 2004. The epidermal growth factor was isolated by Dr. Cohn in the 1960s. But I thought this would be a good time to honor John Mendelsohn, a friend and mentor who just recently passed away, who really is the one who took this into the clinic. I still remember when he brought these agents to us at MD Anderson in 1997, both the antibody and the small molecule. And this is the diagram, this is the slide John used to use as he spoke in those days, talking about how we could target this receptor and how important it was for cancer. Of course, the science progressed incredibly fast. This is why lung cancer is such a scientific field today. We get tissue now in all of our patients. And then, of course, in 2004, the discovery of the EGFR mutation, the fact that we know that there are specific drivers. About 10% of the patients in the US will have this, 20% or more in Asia, hence the reason why we're seeing so many of those patients on these trials. And really let the games begin. It really is so helpful to patients. So this is where we stand in 2019. And of course, drug approvals came rapidly. The first approval of gefitinib, a conditional approval in 2003, but you can see right behind it came erlotinib, first in the second line, then in the front line, gefitinib in the front line, afatinib, and of course, osimertinib. and most recently, dacomitinib. So we have many drugs. When we talk about first and second generation, we basically talk about gefitinib, erlotinib, and afatinib, and then the third generation, pretty much osimertinib, dacomitinib. I'm not sure it's sort of in between. It's the middle generation. The data are quite strong. Here you can see osimertinib outcome in refractory and frontline lung cancer, the AURA3 trial. Here's osimertinib versus platinum pemetrexed. Very strong improvement in PFS, this being in the refractory setting, patients who have had first or second generation drugs. And then, of course, FLORA in the last year and a half or so, osimertinib versus standard of care. We don't have the final survival for that yet, but it's looking pretty good. But the question about EGFR mutated disease, and I see many of you in the audience who have been doing this as long as I have, for almost 20 years, is anyone cured? As good as these agents are, is anyone cured? And I'm afraid the answer is probably no, because of the resistance that develops. So what we're hearing about today, developing new agents to target resistance, is so vitally important. And this is really the paradigm. There's a slide from Jean-Charles Soria, adapted from him, where you have a current standard of care now, would be osimertinib in most places in the frontline setting, and you have a PFS of about 19 months, a response rate of 80%. But undoubtedly, resistance will occur. What are we going to do for those patients? Well, I appreciate this slide from Katie Paletti and her team. And here you can see that you have to think about what is the type of resistance. So we need personalized therapy at resistance. So here are patients who get first or second generation EGFR inhibitors, and you can see the resistance spectrum. Then we were left with about half the patients with T790M. Now, of course, osimertinib was designed to target T790M, and of course, you can see that it was used in that setting. If there was no T790M, we were sort of stuck. We went back to chemotherapy, not very palatable for the patients or for us. And now the c-Met is emerging as a real resistance driver, and you can see it post first and second generation, perhaps even more post the third generation drug. And you can also see it in those patients who are getting the frontline osimertinib. You can see that the c-Met is developing as well. So we have a new target. We have a way to target resistance. So really, how can we get as close to a cure? We have to think about, is the resistance EGFR dependent or independent? Not really clear in today's discussions which that is. It's probably a little bit of both. But clearly, if we find new vulnerabilities of EGFR off-target or independent mechanisms, we can then add to and target resistant patients. Identify genetic or non-mutational mechanisms of resistance, epigenetic mechanisms, for example. Establish the impact of co-occurring genetic alterations, which we've been talking about today. Understand the biology, heterogeneity, and vulnerabilities of the persister cells, the cells that just never die, and determine the interplay between the tumor microenvironment and drug resistance as well. And I think we'll be hearing more later this year about the importance of angiogenesis. And here's from Suresh's paper, or ESMO presentation not too long ago, and you can see in those patients, this is using next-generation sequencing, one of a multiple number of methods that can be used to look at MET amplification. You can see 15% in that study. So high levels of patients, so really an important target to consider. So what about the data? What can I say about the data? We've just had two very complete presentations, and I'll just make a few points. Well, first of all, TATTON was actually a 3-arm study. There was osimertinib plus durvalumab, an IO inhibitor, in addition to the 2 arms that we've heard about today. This arm was closed early, and I'm not going to talk about it. If you want to hear about immuno-oncology, you can go to every other room at this center right now, and you'll hear about immuno-oncology. But I'm going to talk about the targeted therapy and these 2 arms, the osimertinib plus savolitinib and the osimertinib plus selumetinib. Remember, this is early studies. We're in the big plenary room, but these are safety tolerability studies. They're still very early, but it's exciting because it's a lot of patients and it's a very high unmet need. So first, the way the TATTON worked, and I've been studying this now for several weeks, is it was for patients in the second-line setting, advanced lung cancer, EGFR mutant, but really the allocation was by c-Met. So if you were c-Met positive, you went into the upper arm with the MET inhibitor, and everyone else went into the MEK inhibitor arm. So there was selection for the MET arm, but no selection for the MEK arm. And then, of course, the decision was whether or not you had a first or second generation EGFR TKI or a third-generation TKI for the MET patients. And for the negative patients, they were divided up, as you saw, either second line or more, or the T790 negative patients. Let's talk about MEK first. Well, we heard about this from Dr. Ramalingam. The rationale for this combination, we know that upregulation of these pathways, RAS, RAF, MEK, ERK, is a mechanism of acquired resistance to EGFR TKIs and can occur downstream of various other signaling pathways. We know resistance to osimertinib has been shown to be developed because of this signaling pathway in a number of different models and resistant cells. MEK inhibitors can restore sensitivity to osimertinib, and osimertinib resistance by MEK amplification can be prevented by upfront inhibition of MEK kinase, pretending that maybe this could be used frontline. Though remember, there was no selection in this trial. Perhaps that could be added to future studies. Here's the drug, selumetinib. It's a specific inhibitor of MEK1 and 2, MAP kinase. It's oral, and it's been extensively studied. There were large studies in RAS mutant patients with docetaxel that unfortunately, in the absence of selection, did not show any benefit. Here's the patient disposition. I just show this to mention that there were multiple dose-finding cohorts, and it looked like it was somewhat difficult to find a safe and tolerable dose. Doses were different in Asia versus the US. They were using fixed doses in some cases. Bodily size can differ between countries, mean and average size. But it looks like they finally ended up with a dose of an intermittent schedule, 4 days on, 3 days off, because of some of the toxicities that we saw. And here's the toxicity summary from Part A. And just to point out, you can see here that the ultimate doses chosen were the intermittent schedule using the drug days 4 days on, 3 days off. Actually looked a little bit worse here, grade 3, 4 than here, though the DLTs were the same in both groups. Remember that these drugs do have toxicity, diarrhea, rash, fatigue, LFT abnormalities. Not the easiest thing to combine TKIs, of course, because you have both the on-target and the off-target toxicities of the drug. And here's the results, and the results look okay until you realize that most of the activity are in those patients who are T790M positive, patients who have not yet been exposed in this cohort to a third-generation T790M inhibitor. So as I showed you, this group should have a 70–80% response rate already, and those are the blue patients in the first group. There are a few patients who already had osimertinib in pink shown here, or T790M negative in yellow, who had some activity. So perhaps there is a group of patients who might benefit more. Again, maybe a biomarker would help. Then, of course, they went to Part B, and in Part B, the dose of 75 BID, 4 days on, 3 days off was chosen, along with a standard dose of osimertinib. And here are the results here. And again, pointing out, most of the activity, again, small numbers, are in those patients who are T790M positive, though a few more patients now in the T790M negative group having activity. So, you know, my thought here is perhaps there is a biomarker and there is a way to make that number a little bit higher than 23% by selecting patients with a biomarker. And that's something that could certainly be done as these trials move forward. Now let's move to the MET part of the trial. And as I mentioned, MET was divided based on whether or not the patient had a third-generation TKI that targets T790M. Two abstracts condensed into one very nice presentation. There's a lot of data here. I mentioned 15% or more lung cancer patients with MET positivity after treatment, and it could be as high as 30%. This literature is still evolving. It's certainly upregulated post-EGFR resistance. Synergy has been shown for dual EGFR and MET targeting. The paper from Engelman and colleagues in 2007 really was one of the key papers in this field, and it's great to see that this is really now making it in clinical practice. And there are multiple drugs available. The question is, how do you measure this? By FISH, by immunochemistry, by next-generation sequencing, and that, I think, is something that the authors grappled with as well. So in their study, they used a number of different methods. They were done on site and then confirmed centrally by FISH, fluorescence in situ hybridization. So it could either be copy number based by FISH, on-site immunochemistry shown here, and I'm not going to read through it. You already saw it in Alicia's presentation, or it could be done by next-generation sequencing. And actually, there's a very nice poster that I'm not going to talk about because they're going to show it, I think, either tomorrow or Tuesday by Hartmeyer, where all of these different methods are compared. But I thought I'd just say a few words about the methods. So MET amplification by FISH, these are some slides that I got from Zenta Walter and some help from Fred Hirsch, Zenta being our pathologist at Yale. But of course, the MET gene is located in chromosome 7, and what you do here is you look at the centromere on chromosome 7, and you look at the MET gene, and you look for an amplification, and here you can see the centromere is in green, and multiple copies of MET, a very positive result. This is in a right upper lobe lung biopsy, and here you can see in skin metastases the same thing, centromere and upregulation. This is the method we standardly use for this, and we believe to be the most sensitive, though it doesn't get all patients, as you'll see if you go to that poster. Then, of course, you can do IHC. Here's IHC to a cytoplasmic domain of the antibody. David Rim will tell you you've got to make sure you have the cytoplasmic domain if you're using a small molecule TKI. If you're using an antibody, you might want to make sure you check that you have the right epitope in the extracellular domain. But this is a method that can be used as well. And then, of course, next-generation sequencing. This is the ion torrent method. That's actually something that's done at many centers. That method is not as sensitive as some of the others from my research and from experience. Of course, I'm blowing up here chromosome 7. Here you can see the amplification. This is a pretty profound case. But you can imagine if you have an impure sample or you have a small amount of sample, you might miss it. In this study, they accepted all 3. And then, of course, how can I not mention the exon 14 skipping? That, of course, you could do by FISH, or we do it at our center at Yale by the ion torrent, by next-generation sequencing. And, of course, that actually is quite exciting data. And for those who can't remember back 1 hour, here's a little review of that. And it looked like it's quite active, this drug, in the exon 14 skipping patients. So what about the rationale for the combination? Here's savolitinib, goes by a number of names. It's been around for a while. An oral, potent, highly selective MET TKI, which has shown activity, and we now see it has single-agent activity as well in the prior study in exon 14. And here basically is the study we just saw, patients with EGFR mutation positive advanced lung cancer, MET positive status by one of the methods I just mentioned. And then they either had received prior treatment with a first or second generation drug or a third generation drug. And here's the dose that came forward. One wonders if the dose of 600 might be even a bit too high and could be lowered. And from the future studies that were shown, my suspicion is that dose studies will be done to see if the dose could be even further reduced. Primary endpoint, though, safety and tolerability. Well, here are the results, and this waterfall looks quite exciting. Again, early results, but these are all patients who are T790M negative. So you would think that in already being treated with an EGFR inhibitor, there wouldn't be the 60, 70, 80% activity of osimertinib in the combination. And here you can see a response rate of 52%. But equally important is the stable disease rate of 35%. And this looks quite good. Now, I asked a question earlier because I was wondering why these 2 most profound responders were negative, and perhaps it has to do with the assays and having enough tissue for confirmation. And I think that's something that really needs to be done in the future now that we have a target that looks like it's going to be a very valid and real one in lung cancer. Try to understand what the best method is to capture the most possible patients who might benefit from EGFR-MET combo. And then here are the data in the Part B. Interestingly, the results came down here. These are patients with prior third-generation EGFR TKI. So perhaps osimertinib does work again when you retreat. You can see the response rate is 25% only. Although if you look at the stable disease, that's another 44%, and this does look like a pretty positive waterfall. When you looked at the fine print and you looked at the patient's demographics, in this setting, 50 to 60% of the patients had had 2 or 3 or more prior therapies. So it looks like these patients didn't get to this drug and this combo until after they've had, and I don't have the details of this, it'd be interesting to know either other targeted therapy trials or chemotherapy, so they were perhaps a less fit group of patients. But clearly there's activity here, as one can see. A much more heavily pretreated group. And then as far as toxicity goes, it looks like this combination does look to be a bit better tolerated than the MET combination, still with a third of the patients or more having grade 3-4 toxicity. These toxicities, again, being what you see with TKIs, LFT abnormalities, rash, fatigue, some pyrexia. And again, the question might be, could the dose be reduced a little bit? And again, this was mostly an Asian population, so perhaps the dose needs to be reduced in Asia or could be reduced in other rest of world populations, as they call it. Now, in the last few minutes, what about, or what can we say about outstanding questions? Well, there are a couple of questions I have for these authors. One, what about the composition of components? We have doublet regimens here. What is the single agent activity of MET and MEK? And that, of course, will have to come out of randomized trials. I think there are preclinical data to suggest that you should probably use the EGFR inhibitor with them, but certainly it would be nice to know a little bit more about the single agent activity of both. Do the lines of therapy make a difference? We certainly saw that between the two groups. MET inhibitors used in the last line of therapy might not be the most effective place. Over the years, we've learned in lung cancer therapy that moving our best agents up forward, especially if there's a target that's validated, is probably the way to go. So frontline trials might be warranted for both of these combinations, perhaps. And then the need for more profiling and selection. It's still going to be important to find something for MET that can be the most inclusive, but also allow for the best result, given that when these drugs don't work, they cause toxicity, so we only want to use them in the right patient at the right time. For the MEK inhibitor, clearly there's a population that's benefiting. It has some activity in some setting, in my opinion, but it needs to be honed down and found. Given the toxicity, biomarker work should be done in future trials. So finally, what is the impact of these data on clinical practice? What are the next steps? Well, I tried to put the results, onto the diagram that the authors used for the MET positive and the MET negative patients. On top, we have the MET positive, the patients who got the savolitinib, and you can see the response rates, both for the EGFR prior third generation EGFR TKI, or those who received prior first or second generation EGFR TKI. They did not show us today patients who were T790M positive. But I think what we see is that the response rates are reasonably good, and the stable disease rates quite high as well. The grade 3-4 toxicity, acceptable, and the durability of response, which I'm not showing here, but we saw over 7-8 months, quite good. For the MET negative patients, numbers are a little bit smaller. Certainly in the group that had prior first, second generation, small numbers, the response rates are high, but again, a lot of that could just be targeting T790M. For those who received prior third generation, modest results with significant toxicity, so this might need a little bit more study. So how will this impact clinical practice? For MET positive, I would say this is impressive early data. We need randomized trials to confirm the results, need to better define MET selection criteria, and this could, in the future, be a very useful refractory EGFR therapy in a world where we're all, whether you're in Asia, Europe, United States, any place on the planet, we need to find better ways to treat these patients when they become refractory, and this looks like a big piece of the pie. For the MET negative, data are even earlier. Toxicity seems manageable with a non-continuous dosing. It's not the easiest type of schedule for a patient to follow, but it's doable. Difficult to assess efficacy in the mixed population. There's a possible utility up front, perhaps with a biomarker or using it up front. And genetic profiling, I think, could be helpful. This was basically everything else, so it would be nice to have some more biomarker work there. There are a number of trials planned. We heard about this. Savannah, very appropriate given where we are today. Osimertinib, advanced EGFR mutant lung cancer, a refractory trial looking at the combination, and you can see they're looking at 300 and 600 milligram dosing, so perhaps going down in dose a little bit. Then there's this trial for osimertinib in combination with selumetinib, an EGFR inhibitor naive advanced patients, so moving into front line at the dose level discussed, and this Orchard trial, which is going to be a battle-like design to sort of personalize therapy for this disease. So I promised Dr. Yap, who gave me this title, that I would answer the question, so let's do it. Should we get a tattoo? So no one could tell me what tatten meant, so I said tattoo. Mother's Day's coming, so my team helped me with this slide. So which of these should we endorse right now? MET or MEC? Have we met it or have we MEC'd it? And I would say, given what we've seen today, both have some activity, but today, the third day of the baseball season, I would say we've met it. Thank you very much.
Great. Thank you very much, Roy, and thank you all for staying to the end. I'll now bring the session to a close. Thank you.